So my name is Emily Evans. I'm from the University of Minnesota, and I'm going to talk to you today about a project that we've been working on for the last several years, and it's looking at how tillage radish can complement organic rotations. So the research was done at the Southwest Research and Outreach Center, and it's one of 10 research stations that we have across the state. Ours is in this little bottom corner right there. The research was done. We have a 120-acre piece of land that had pretty much minimum inputs its entire life, but it's been certified since 1998. And so how this came about was, in our area we've got really dark, heavy clay mollisols, and a lot of the farmland that we have is poorly drained. And so a lot of the conventional farmers in our area have recently expressed a big interest in tillage radish, and the reason they're using tillage radish is to break up subsoil compaction. So there's been a lot of talk, there's been a lot of aerial seeding of tillage radish into standing corn, which in our area, we have a very rural area, but what that also translates to is now we have a lot more availability and access to the seed. So organic farmers were becoming a little bit more interested in how this could work in their rotation, but they were looking at it more from the perspective of nutrient recycling standpoint. And they also were interested in the weed suppression, but they really expressed a lot of interest in that nutrient scavenging part of it. And what makes tillage radish, I don't know, has anybody worked with tillage radish here? Has anybody grown? So then the cool thing about tillage radish, and there's a picture right here on the bottom here, and that actually is a little hole after it decomposes. It's got a super, super strong taproot. It can go feet deep, inches wide. It can break through that subsoil compaction, and then when it decomposes rather quickly in the spring. And when it decomposes, you can actually go out there. Now, usually I have my foot in there, and you can see how big the hole is. It leaves holes just through your crop. It's actually really cool. But what's even cooler about in our area is in the springtime, we have lots of rain and snow melt, and farmers are kind of getting anxious to get into the fields, and so this actually helps it warm up and dry out, and especially organic farmers when they have when are hardy crops or perennial crops and they have that extra added task to terminate it and work it. It kind of gets, I'm a farmer's wife, you get a little anxious, and so it's just kind of a nice added attraction. And the fact that it's fast growing too also helps because it can fit into those short rotational windows, which we definitely have in our area. And so when we talk to these producers, their concerns lie in two areas. And one was that the ability for it to recycle that nitrogen. And so the ability to scavenge is very well documented. There's been lots of research on it. But the timing of that nitrogen availability to that following crop is really inconsistent. And it's not even that it's understudied, it's that it's really hard to pinpoint where that nitrogen is going. So we really wanted to focus on that. But the producers also had other questions about best management practices, specifically a planting date and then the fertility needs of tillage radish in order to get it established. So for planting date in our area, it was pretty much agreed upon that mid-August date was a good point. So we decided in our experiment that we were going to follow a small grain with tillage radish and then follow it the next year with a corn crop. But we also wanted to put in a manure component so we could test that fertility need and see if we really do need some extra added nitrogen to get that crop going, that cover crop going. And so we set up an experiment, randomized complete block design, 4 replications with 3 objectives. We wanted to look at system effects, and when I say system effects, this is that manure component. So we added that manure component in 2 out of the 3 treatments. We wanted to look at planting radish by itself, but also planting it in polycultures to see if we can extend that nitrogen availability. And then, obviously, we wanted to see how it impacted our corn yields. And so these are the experimental treatments. So on the left side right here, you're going to see these are cover crop treatments, and over the top, that's what we called our system treatments. And so with the cover crop treatments, first we planted tillage radish by itself. Second, we planted tillage radish with raceme clover and annual oats. And both of these do not survive the winter in our area. So we just kind of wanted to put this in to see what the effects of planting cover crops with different types of residues and the different rates of decomposition, how that would extend the nitrogen availability. And then we included this last one, so it's tillage radish, berseem, and winter rye, with the idea that that winter rye would continue to grow. It is winter hardy in our area, so in the springtime it would continue to grow, and as the tillage radish decomposes rather fast, we wouldn't lose that nitrogen, but we could rather re-scavenge it or use that winter rye to recapture that nitrogen that's thought to possibly be lost. And then when we terminate that and plant corn into that, we have an extension of that nitrogen. And then on the top up here, these are our system treatments. And basically when we say system, the only difference was how the plots were treated prior to cover crop planting. And so that first one, we had no manure, no tillage, we just direct seeded, we no-till drilled it right into the wheat stubble. The second one, we did apply manure on the surface, but we didn't do any kind of tillage, we just direct seeded it right into the stubble on top of the manure and let the manure lie. And then the third one, we did some tillage, but it was just a very light tillage. It was just to work the manure into the top for probably 4 to 6 inches. And then we planted into that. And so this is just an idea of the operations. We repeated this study from 2014 to 2017, which ended up being 4 years of that wheat cover crop phase and then 3 years of the corn phase. And so you can pay attention right to this cover crop planting. You can see in 2014, we didn't hit our target date. We were middle of September instead of middle of August. And this is a function of not having funding. And so then, you know, your project gets kind of put on the back burner. But you might see the effects of that in the next couple of slides. And then the 2015 and the 2017, that was planted August 24th and 25th. And that was because we had a really wet August. We usually have a typically dry August. We didn't see much of a decrease in the cover crop. It's probably because we had really nice soil moisture when we planted it. But that kind of told us, you know, if we have these results over 4 years, that we might have a little bit more flexibility in that target planting date. And so the samples that we collected, we looked at cover crop biomass. We did it at the end of the cover crop growing season, and then we also took biomass samples of the rye regrowth in the spring. And in the soil, we tested the soil in 2 points in both years. So we took it before cover crop planting and before winter kill that first year. And then in the corn year, we did it 4 weeks after the corn planting and then after harvest. And the idea of taking it at that 4-week mark is that's thought to be when corn really kind of ups its game and starts to really accumulate biomass, and so that's when its nutrients needs start to get up. So we wanted to get a snapshot of what was going on in the soil. So what did we find out? So what we're looking at here is the cover crop biomass. And remember what I said about that 2014 planting date? So this is 2014 and this is 2014. So along the bottom, those are the years. Year 1 is 2014, 2 is 2015, and so on. And so you can see in 2014, we didn't have as much biomass as the other years, and this is very apparent visually in the field. So on the left-hand side, the left figure, that's just the tillage radish biomass. And on the right-hand side, that's total biomass. That's including the berseem, clover, and the winter rye. But what this is actually showing is this is the system effects. We didn't see any cover crop or system interaction. So we just looked at the system effects and the cover crop effects, and this is the system effects. And so just to recap, we see this up here is the legend. That's system 1 and system 2, that red and that brown. The only difference is that system 2, the brown one, got manure. They were both no-till drilled. And we consistently saw no difference, which tells us that we didn't really see that manure helped, promoted, did anything with the tillage radish growth. Now, what's interesting though, is we see between these last two, system 2 and system 3, where system 3 is in the yellow, that's the one that got the tillage. So they both got manure. The only difference is that system 3 had that tillage. We saw significant biomass accumulation, higher. And talking to some farmers, they're like, oh heck yeah, that's what we see too. And so it's not that you can promote tillage, but there are definitely times and places. It's smart tillage, or it's advantageous tillage, it's mindful tillage. And so that's just what we saw in these. And visually, when we were in the field, we saw this too. You could walk through the plots and you could tell what was planted in that third system. And so we didn't do any kind of data collection with the below ground biomass, but we did observe something similar. So right here, this treatment 5, treatment 8, and treatment 11, that was that third system treatment that was before tillage. Now this was in 2016, which we had a little bit less biomass compared to 2015 and 2017, but you can still kind of see the differences. And ironically, this right here, which says A, that was the tillage radish that was in our alleyway, which we tilled at the same time. And didn't even have, we didn't even apply manure to that. So it really showed that we saw something with that tillage prior to planting. And so in terms of cover crop biomass, as it was affected by the cover crop treatments, we didn't see that tillage radish had any growth effects when it was planted with a polyculture, which we can kind of infer then that the scavenging capability is very similar when it's planted alone within a polyculture or compared to within a polyculture. We didn't change the seeding rate. That was one thing because our objective was to get it to scavenge that nitrogen, and we wanted to see where it would go. So I suspect that possibly we'd see different results if we reduced those seeding rates. So, since we're talking about tillage, I thought we should probably be looking at the percent cover. I wanted to see how much these soils were covered over the winter since we were doing a August or September tillage. And so this is what I think is kind of interesting. So this is the average between 2015 and 2017 over the years. And this top bar right here, that's that third system again. So that's where we did the tillage. And this here is the second system, and that's here the first system. And so the green is the actual living percent cover. And of course, I mean, you saw from the pictures of the biomass, it makes sense that it has about a quarter to a third more than the other two systems. But what I think is really interesting is what all three have in common is the percentage of bare soil. So, and this was really consistent over all the plots. They were all extremely similar. In fact, they all had between 86 and 89% cover. So the real significance of this, and it's not just for organic farmers in our area, but in our area, and I'm not sure how it is in other places, but we have many farmers that tend to like to turn the soil black in the fall. Yeah, and the reason behind this is because they're thinking of that springtime, and you know the black soil is obviously going to warm up much faster and dry out much faster, and they can get in the field. But when we're looking at the soil cover and we're looking at the potential of tillage radish to actually do that same function, that's really a tool that could be used, not just in organic, but in conventional as well. So the soil results. They're very disappointing. So these are the averages in the corn samplings both July and fall. And so we looked at the extractable or the available nitrate and the available ammonium, and we also looked at mineralizable ammonium, and what we found was nothing. We saw no differences in treatment, we saw no interactions, we saw no cover crop effects. So it was a little disappointing. We wanted to see something, but sometimes when you see nothing, that is something. So it means that we have to look a little bit further. We also looked at the soil biological activity because we're thinking, okay, something's got to give. Something has to be different. We looked at 4 different enzymes. We looked at permanganate oxidizable carbon, which is correlated with microbial biomass. And it's kind of an indicator of that active carbon portion. And we also looked at, again, the mineralizable soil ammonium as an indicator of activity. And again, we saw nothing. We saw no consistent effects of either cover crop or system. So that was really kind of surprising to me, especially the oxidizable carbon, because that's usually pretty sensitive to tillage and to additions of a lot of biomass. So yeah, that kind of threw me a little bit. So now on to corn grain yield. That's my daughter. She's glad that she got to be part of the slideshow. And so this is the statistical analysis table of the corn yield. And so we didn't see interactions in any of the years. But we did see, in 2 of the years, we saw cover crop treatment effects. And in all 3 years, we saw pretty significant system effects. So, looking at the corn grain yield, again, the years are on the bottom. So this is 2015 in year 1, 2016 and 2017. And these are the cover crop treatments. And so we see that winter rye, and we can pretty much explain this we think. The winter rye had that third treatment is the winter rye treatment. It had a negative effect on the corn yield. And I think this was definitely a management problem. We had wet springs, it was hard to kill. We left a 2-week window, in fact, sometimes even more before turning it in, planting corn. And I think it was just, frankly, too mature, and we had some nitrogen tie-up. But this is what's interesting. Again, we saw very similar results as we did with the cover crop. So we see that system 3 treatment, again, had a positive impact on the corn yield. And so, in conclusion, we did see that that tillage component had a positive impact on the cover crop growth, and that translated into the corn yield. We don't have any information that had anything to do with nitrogen, which makes sense because we know that tillage radish really does change the physical properties. So I'm just, for the sake of time, because I know it's time for questions, just to move a little bit further, I think it's worth researching more into the soil properties, because I think it's really important that if we see something happening, that we can really understand exactly what is happening so that we can exploit those characters and find where it fits into our systems. And that's easier to translate across regions when you actually know what it is that you're planting that cover crop for. The other thing I'm really curious about is where that nitrogen is going, because this has been similar through studies that have been done in Wisconsin and another one that was done in Minnesota. And one of the researchers even said to me, I don't know where it is. I don't know where the nitrogen went. So I think it's kind of our responsibility to find out, and I'm curious to actually see if it's volatilizing, because that environment in that hole is very conducive to volatilization when it's warm and it's moist in the spring, especially with a lot of rains. And of course, other entry points for this cover crop. We now have an interseeder that we can seed into corn up to the I think they interseeded into V12. They're doing this on the conventional acres, and we're going to start doing it on organic. So it's just another tool that we can use to get cover crops in different parts of our rotation. And with that, I thank you, and I will take any questions.
What were your manure application rates?
So we applied by hand, but they were equivalent of 4 tons per acre.
You have a question?
Yeah, I'm just curious, what depths were you looking at the carbon when you were?
Just the top. Yeah.
Right.
Any of the biological, we only did in the top 6 inches.
Any other questions? Oh, one over here. I'm bringing the microphone.
And also with the nitrogen, we actually went down several depths, but I only presented the top, but we didn't see any differences there either.
The length of the no-till radishes looked very disappointing in the pictures that you have there. And so what do you think of the stimulation behind the tillage that improves the size of the radish so much?
It could be several things. I think this seed to soil contact is a huge thing. It kind of fluffs it up, the aeration. I don't know, it could be several things. I'm not quite sure. It might also have to do with a little bit of moisture, because if we're only putting it into the top, and that time of year typically is pretty dry. And so if we're doing some tillage, we're stirring that all up and we're bringing that moisture from down below, and we're planting it pretty darn close to after we do that tillage. So I think it could possibly have something to do with just a little bit of increased soil moisture.
Yeah,
no differences in your soil analysis, and I'm wondering because sometimes the method that you're using for the analysis is really important. So what kind of method did you use for your soil test analysis?
For the nitrogen,
Right.
so we did an incubation method, and then we did a extraction, and then we used a spectrophotometer. Can you ever say that? Yeah, and so we did a KCl extraction, and then we used the vanadium chloride for the nitrate. Have you heard of that before? And then the incubation was actually a 7-day long incubation, and then we did the similar extraction to get the ammonium. The vanadium chloride was for the nitrate. We used a different method for the ammonium. And this is something I've run on several other. I mean, we've run it on everything, so we've seen. And it's not that we didn't get nitrogen. We got nitrogen, but there was just statistically there was no differences. So we saw that there was an increase in nitrogen in certain stages, but it was the same throughout all the treatments.
We have one question in the back. I think we have time for one more.
Hey, great presentation. It was interesting to hear that there was no differences in the active carbon fraction. Are there other fractions of carbon that you would incorporate into future studies, sort of to use as indicators of future carbon changes that you know of?
I don't know. The active carbon is nice because it's more of an immediate. It's supposed to be that's the other thing. So after we didn't get that, we extended each one. We actually did plant soybeans after corn. And we treated every plot the same, so we just put in one more year. We kept the plot separate, and we did another sampling. And so we thought, okay, we'll run these biological indicators again because something's got to be there. It wasn't there, so it was just a little frustrating. And I mean, for many people, and I'm sure you understand if you do research, it's all about time and it's all about money. And so it would be so nice to look at several points. You just sometimes with you're restricted and you have to try to pick the best points, but I kind of wonder if maybe if we'd go back and did it on more of a monthly, did 5 different samplings and really try to pinpoint that. But I was really surprised with that, the active carbon part of it.
Great, thank you so
Yeah,
much.
thank you.
Emily.